Welcome everyone to my PhD seminar about microbial production of bio-based chemicals, a biorefinery perspective. My name is Tariq Shisha, and I'm going to take you a small trip to chemical industry. So first, overview of the presentation. We have, I will give a short background about chemical industry, biorefinery, and industrial biotechnology, and then move to the objectives of our work, which is microbial production of chemicals, and we have a list of chemicals here. And then I end up with concluding remarks and future perspective. So, first of all, what's the chemical industry and how big is this market? This market now, in 2002, the estimation said that it exceeded 1.3 trillion US dollar, and now there is some, it could even exceed 3 trillion US dollar. Almost 90% of the chemicals that we get nowadays, it comes from petroleum and natural gas refinery processes. However, because of these processes are finite sources, so there is a risk of depletion, and at the same time, the price of chemicals in the market of oil is increasing, and this led to increased prices of a lot of chemicals and a lot of things in the market. As well, in chemical industry, there is some toxic raw materials, toxic intermediates, and also some processes have a bad environmental impact that led to greenhouse gas emissions, and this led to many people start being more aware about the environment, and they start asking for green products. And as well, also they start asking for the carbon footprint of different chemicals. So now the trend is to go to produce these chemicals from renewable resources. And we can see here, renewable can be, we can get renewable energy or renewable chemicals and materials. For energy, there is many alternatives, not only biomass, such as solar and wind and water, energy from water. However, for chemicals and materials, the main source is a biomass. Biomass is not only plants, it can include any animal byproducts, human or animal waste, and many other things. In order to convert the biomass to chemicals and materials and fuel, we need a facility, and this facility is known as biorefinery. And in this facility, the biomass conversion process is integrated with equipments for conversion of these chemicals to the end of this biomass to the end chemicals and materials and power and so on. And when it started the biorefinery, there was the first generation of biorefinery, and we can see here that it started with production of biomass, and later on processing and producing biofuel, which being used and release carbon dioxide to go back again to the nature. But this resulted in many concern about the land use for production of biomass, and also it was not economically feasible because biofuel is not an added value to this process. This led to the second generation of biomass, and second generation of biomass, they start focus more, second generation of biorefinery, and in the second generation start focusing more on biomass waste, not the biomass itself. And the main focus was the production of chemicals, materials, and then that go back for application and biodegradation in the nature back to carbon dioxide. And side stream can be used for production of biofuels, and by this way you can add a value to the biomass. So now we will have two things, biomass, and we want to convert it to fuels, chemicals, and materials. And there is four different concepts. The first concept is to make a direct production of the desired structure directly in plants. Second concept is to do one step of biological or chemical modification of naturally produced structure. Third one, to make several processes of modification. And finally, to break down the biomass into small units such as carbon monoxide and hydrogen in case of syngas, doing pyrolysis to get a bio-oil. And all of this can be combined later on to get the chemicals that we want to produce. In our case, we are focusing on several step modifications of naturally produced structure. However, in order to develop a biorefinery, we have to understand or to know more about the petrochemical industry. In petrochemical industry, you start with natural gas or crude oil, and it go for refinery and processing, further on to steam cracking and catalytic reformation. But the most important here is the this first line of chemicals, which is around 6 chemicals, and this represents the starting material for most of the chemicals and the materials that's present nowadays in the market. So several chemical processes, you can reach different products that you want in the market. In our case, we were studying the ones that are colored, with the colored boxes here. We were trying to produce these chemicals from industrial and agricultural byproducts. So taking this as a model, we go to the biorefinery. In biorefinery, we have starting materials from a biomass, and then do some processing, so you get the number of carbohydrates and number of sugars and so on. If we focus on one of them, which is a glycerol, because we used it in our process, glycerol comes out from the methanolysis of vegetable oil for production of biodiesel. That's used as an energy carrier. And it's accumulated as a byproduct, and that's why we won't use it for production of chemicals. Glycerol was found to be, it can be converted chemically and biologically to a wide range of chemicals. So it's considered as a central point that you can produce a lot of chemicals similar to petroleum refinery. In our case, we were studying the conversion of glycerol to propionic acid, 3-hydroxypropionaldehyde, and 3-hydroxypropionic acid and 1,3-butanediol. Also, it's not only glycerol that's central metabolite. 3-hydroxypropionic acid is considered a platform chemical that you can convert chemically or biologically to a lot of other products. One of the most important is acrylic acid. Also, poly 3-hydroxypropionic acid, that's a biodegradable and biocompatible polymer. And also, 3-hydroxypropionic acid have many applications as a in cosmetics and in food and many other products. Second one is propionic acid, that's used mainly as a preservative in feed, food, and pharmaceuticals. But also it can be integrated in cellulose acetate propionate plastic. It can be used in flavoring agents and also in herbicides. 3-hydroxypropionaldehyde, it has an antimicrobial activity that allow it to be used for industry, for food industry and the healthcare sector, but also can be converted to a wide range of chemicals that can be produced in the market. And by this way, you can have a new central metabolite, compounds that you can convert it to a wide range of chemicals. And in order to achieve this conversion from glycerol to these chemicals, we used industrial biotechnology, and it deals with the use of microorganisms and enzymes as catalysts in a process such as fermentation and biocatalysis. And in many cases, the industrial biotechnology was shown to be more cleaner, energy efficient, and less toxic than chemical process. However, we need some indicators to know if the industrial biotechnology, if the process that you are developing is okay or not to go for industrial scale. And it includes some factors such as volumetric productivity, which is the amount of product produced per unit time. And it affects mainly the reactor size. So for example, if we have low volumetric productivity, we'll have certain reactor size. Increasing the volumetric productivity, we can reduce the reactor size on a larger scale. Also the product yield, which measures the amount of substrate that's converted to the desired product. And that affects mainly the amount of raw materials that we want to use. So low product yield will give certain amount of biomass that we need, and increasing the product yield, you can lower these raw materials. Third is the product concentration, and it affects mainly the downstream processing for purification. So if you have a low product concentration, you have to handle a large volume of water. Increasing the increasing the product concentration, then you can handle lower amount of water, and all of these three factors will affect the process economy. Now I'll go to the objective of the work. So we have aiming to develop economically competitive process for production of chemicals based on biotechnology, and we started with glycerol that can be obtained, or even in some cases we use biodiesel-derived glycerol. And we want to convert it to these chemicals, which is a platform chemicals. By fermentation, by transformation, that's the two processes that we use in our study. The second aim is to integrate biotechnology with chemical process for production of secondary chemicals, and in this case we use 2-methyl-1,3-butanediol, that's an industrial byproduct, and by biological and chemical conversion we can get methacrylic acid. So, going for the work. First compound that we deal with is propionic acid, and we studied it in paper number 1 and 2. This propionic acid have a very interesting, we used propionic bacteria for production of propionic acid, and this propionic bacteria have an interesting metabolic pathway where you can convert different carbon sources into propionic acid through pyruvate as a central metabolite, and also this pathway pass through succinate. We have some byproducts such as acetate and succinate and N-propanol that are formed also beside propionic acid. And the degree of reduction of the carbon source at the beginning, the starting raw material, will affect the type and the amount of byproducts that's being formed. In case of glycerol, because it have a higher degree of reduction, so you can get higher amount of propionic acid and lower amount of byproducts. So what's the problem of propionic acid fermentation? That's a graph showing a batch production of propionic acid. We have glycerol that's being converted to propionic acid. And we can see here that there is 3 main phases. The first phase is slow glycerol consumption phase. So it lasts for almost 30 hours. You have no glycerol consumption. And the second period is at the end, you have a slow glycerol consumption phase also due to the propionic acid inhibition. And only the efficient period is here, it's quite short if you compare to the whole process time, and as a result you get a low volumetric productivity of around 0.2 gram per liter per hour, which is 10 times less than the requirement for industrialization. In order to avoid these 2 drawbacks, we studied high cell density fermentation. And in order to do this, we used 2 techniques, cell immobilization and cell recycle. In the first case, we immobilize the cells on 2 matrices. The first is called loofah, which is a plant, and it has a legocellulosic fibrous structure, as can be seen from the scanning electron microscopy. And the second one is Poraver, which is a recycled glass beads. And we can see that it has a large number of pores on the surface that can adopt microorganisms. However, the initial trials for immobilization, it was successful, but it didn't get high cell density to be immobilized there. So we decided to treat the matrices with polyethyleneamine, which is a polymer that have a positive charge. And this polymer will interact with a negative charge on the cell surface. And as a result, you can increase the absorption capacity. And we can see here, as a result, we can see here the cells immobilized in case of loofah. And in case of Poraver also, we can see that all the pores are filled with huge amount of cells. And when we start comparing these two, we found that the Poraver immobilized 30 times higher amount of cells than loofah. So we thought, okay, we can expect high increase in the productivity. So going for production of propionic acid using these 2 reactors, we found in case of loofah that the volumetric productivity was constant around 0.3 gram per liter per hour. So there is no advantage over batch process in this case. However, in case of propionic acid and in case of Poraver, we found that the volumetric productivity increased from batch to batch and reached around 0.86 gram per liter per hour, which is 3 times higher than in case of loofah and in case of free cells. However, comparing to the high amount of cells or the big difference between the 2 reactors, we expect that it could be higher, but we found that it could be a matter of mass transfer limitation. You have so much cells and the nutrition is not efficient to reach to all of these cells. And then you have lower productivity. In order to avoid this, we decided to go in the second reactor with cell recycle, where we use the free cells in a sequential batch mode of operation. And generally we start with a normal batch process, and then at the end of the batch we do centrifugation. We send the cells back to the reactor, and the supernatant will be sent for downstream processing. So the cells go back to the reactor and we start the next batch, and so on. This will be repeated several times, and we did it for 11 batches. In order also to make the process more economic, we decided to use potato juice instead of yeast extract. Potato juice is a byproduct from potato starch industry, so it's quite available at cheap prices, so it would be interesting. So we use potato juice in this process, and here is the results. As we can see in the first batch, we have the fermentation time. It takes around 150 hours. while in the last batches it was almost 21 hours. So we decreased, or we cut 125 hours from the fermentation time. And also we remove the time that's required for cleaning and sterilization in place. So we have a batch followed by a batch and so on. And as a result, the volumetric productivity increased from 0.2 to around 1.4 gram per liter per hour. Also, we studied the effect of the optical density on the volumetric productivity. So we found that increasing the optical density by 10 will result in increase in volumetric productivity by 0.2 gram per liter per hour. And also we found that this process is highly stable and we can follow the change from batch to batch. And we found that the increase from batch to batch in the volumetric productivity is exponential, so we can predict the next batch how it's going to increase. And at the end it becomes stabilized around at a productivity of 1.4 gram per liter per hour. Finally, we evaluated the product yield and byproduct yield, so see how stable this process, and found it's highly stable. And even in the last two batches, we reduced the amount of nitrogen source to the half, and we found that acetic acid and succinic acid concentration was decreased by 40%, which increased the purity of propionic acid as well. So to summarize the work of propionic acid that we have done so far, we have here the propionic acid. We decided to choose some parameters and evaluate the results comparing to the results that have been produced in literature. For only glycerol and using wild type or mutant-free or immobilized cells and batch or feed batch operation. And we can see here that there is two main limitations. Either you have a very high concentration of propionic acid, but the productivity is quite low, is around 0.03 gram per liter per hour, sorry. Or either you have a good productivity, but the concentration is too low. And these are too far from the industrial target. When we did the first process for immobilization, we found that we can improve the productivity and the concentration a little bit. When we did the second round for cell recycling, we found that we can also come closer to the industrial target. And even we did more studies, but it's not shown in the thesis, and that we can reach even higher concentration and productivity. So now is to close this gap between what we have achieved and industrial target, and I think we can use a combined technologies, not only a single technology. Second compound is 3-hydroxypropanaldehyde, and this compound, we use Lactobacillus reuteri for production of this compound. And we can see here that this is a metabolic pathway of Lactobacillus reuteri. It's a heterofermentative lactic acid bacteria, so it produces lactic acid, ethanol, and acetate from different sugars. However, here we cannot see the glycerol and the glycerol metabolic pathway, but it's present on the side as a side pathway, not related to the microbial growth. And we can see here it's coded by bioprocesses, and glycerol is converted first by the first enzyme glycerol dehydratase to 3-hydroxypropanaldehyde, which is reduced to 1,3-butanediol and used, 3-HPA is used as an electron acceptor in this case. Also, the microorganisms have a side pathway for oxidation of 3-hydroxypropanaldehyde to 3-hydroxypropionic acid. And the three molecules, 3-hydroxypropanaldehyde, 1,3-butanediol, and 3-HP are industrially important chemicals. So first we'll start with the 3-hydroxypropanaldehyde. Microbial production of 3-hydroxypropanaldehyde have been tested using two methods, growing cells or resting cells. In case of growing cells, you have the metabolism of sugar generates a lot of NADH, and as a result you get high amount of 1,3-butanediol. In case of resting cells, there is no generation of NADH, so most of the glycerol will be converted to 3-hydroxypropanaldehyde, and based on this one, you can get higher volumetric productivity, higher yield, and higher purity of the product. However, there are some limitations, including high toxicity of the 3-hydroxypropanaldehyde to the biocatalyst, which result in inactivation of the enzyme after 2 hours. So the process is almost economically unfeasible. In order to solve this problem, it's possible to do complexation to close this aldehyde group through a reaction with semicarbazide, so you get an intermediate, and then by removing water you can get the semicarbazone, which is highly stable. And here you don't have the aldehyde group free anymore, so you can produce more of the aldehyde. However, to go back again to this reaction, there is no report so far for 3-hydroxypropionic acid that you can, propionaldehyde, that you can go back again to the free form of the aldehyde. Another alternative is to use bisulfite adduct, which is after producing 3-hydroxypropionaldehyde, you can react it with sodium bisulfite, yielding this adduct. And we have studied the formation of this adduct, and we found that it's an equilibrium reaction, so it's only 50% of the aldehyde is converted to the complex. Later on, as this molecule is ionized, so you can bind it to an ion exchange resin, and later on you can elute using sodium chloride to get the aldehyde and the adduct. So it's a reversible reaction, so you can get your product quite pure. So we evaluated we measured the dynamic and static binding capacity, and we determined the breakthrough curve. And also we did a column separation through loading and washing and elution. And we can find that we managed to elute 100% of what is bound to the column. And here we can see the purity of the final product. It's highly pure. We don't have 3-hydroxypropionaldehyde, and we don't have glycerol comparing to the initial solution that we started with. So the next step now is to go for in situ recovery of the 3-hydroxypropionaldehyde. There is many constraints, such as the high toxicity of the 3-hydroxypropionaldehyde to the cells, and also glycerol and sodium bisulfite and the ion exchange resin have inhibitory effect on the glycerol biotransformation. In order to avoid this, we have decided to go for each parameter separately, and we can see here the productivity from a batch, the amount of 3-HPA produced, the amount of biocatalyst, the gram of biocatalyst, in case of batch process. So the first modification, we did the feed batch operation, and we found that we can increase the lifetime of the cells up to 5.5 hours, and also we can increase the amount of 3-HPA produced per gram biocatalyst to 1.2 cells. So the next step is to start adding or making in situ complexation, so we feed glycerol and sodium bisulfite, and we found that also it increased to 2.74 grams 3-HPA per biocatalyst. And finally, in order to shift the reaction further, we connected the reactor to a column for ion exchange resin column for catching the complex and the free aldehyde. And we can find also that we managed to increase the amount of 3-HPA to around 27 grams. And the cells is still active even after 19 hours of operation. Next compound was 3-hydroxypropionic acid. And there is two strategies for production of this compound that have slightly been evaluated, either using wild-type microorganism or using an engineered microorganism. So far, all the studies that have been done on production of 3-HP was successful using engineered microorganisms. Most of the trials on using wild-type microorganisms were not successful. However, we decided to go with the wild-type microorganism and investigate more why it's not no results, and to prove that it's possible to use a wild-type microorganism. In this slide, we show different metabolic pathways that have been suggested for production of 3-hydroxypropionic acid, either from glucose or glycerol. So the first, from glycerol to go 3-hydroxypropionaldehyde, and through different ways you can go or reach 3-hydroxypropionic acid. Also, it's possible to convert glycerol to glyceric acid and malonate semialdehyde, and then later on end up in 3-hydroxypropionic acid. From glucose, you can convert it first to glycerol, that can go end up in 3-hydroxypropionic acid. You can go through lactic acid. You can go through malonyl coenzyme A or alpha and beta alanine. In our case, we investigated this pathway where we convert glycerol to 3-hydroxypropionic acid through this coenzyme A and phosphate as intermediates, and the co-production of 1,3-1-diol in order to achieve the redox balance. So we did co-production of CHP with 1,3-butanediol using resting cells of Lactobacillus reuteri, and we can see here the metabolic pathway for Glycerol conversion. And there is 3 different scenarios. The first scenario is that we have done before for production of C-hydroxypropionaldehyde. You add so much Glycerol, and as a result you accumulate so much 3-hydroxypropionaldehyde, and this aldehyde is inhibitory for the other enzymes, and you end up in 3-hydroxypropionaldehyde as a main product. If we go with a lower rate of glycerol feeding, we can go for scenario number 2. We start feeding glycerol at a lower rate, which allow most of the aldehyde to be converted to 1,3-butanediol and CHP, and a very little amount to be accumulated as a 3-hydroxypropionaldehyde. And using this technique, we can measure the different kinetics for the conversion of 3-hydroxypropionaldehyde to the 2 different products. And finally, scenario number 3, we start feeding at a much lower rate, where it allows complete conversion of the aldehyde to the 2 compounds, and we have no accumulation of the aldehyde inside the reactor. Also, we evaluated resting and growing cells in a continuous mode of operation, just to make a comparison and to prove that it's possible also using growing cells. And we can see here comparison. In both cases, we can achieve 100% conversion. In case of growing cells, you have 1,3-butanediol is the dominant product, while in case of resting cells, you have equimolar of the 2 compounds. Volumetric productivity is higher in case of growing cells compared to resting cells. Here we can see HPLC chromatogram showing the byproducts that's formed also. So we have lactic acid, we have succinic acid, we have acetic acid, and many other products also formed. Compared here to only 2 products are formed. And this for sure will make downstream processing much easier. And we can see also the color of the 2 solutions here, what's coming from the first process using resting cells, and here what's coming out from the growing cells. Finally, I'll go to Yes, that's what you can expect if you have a normal glass. But if you are using polymethyl methacrylate, which is a polymer of methyl methacrylic acid, it's 4 to 8 times stronger than the glass, and also it's have a good transparency and lighter than the glass, and that's why it's used in many applications, and it have a global market of 4.1 million tons per year. So it's a highly demanded compound, and if we look to the current production of methyl methacrylic acid, we found that it's acetone cyanohydrine process, where you react acetone with hydrogen cyanide, methanol, and sulfuric acid, and you get methyl methacrylate. However, this process, if you can see it, it has a highly toxic raw materials with a lot of nice signals, and also you accumulate 1.5 ton of ammonium bisulfate for each ton of methyl methacrylic acid. So even the byproduct is more than the product that you achieve. Not only this, but also you have the carbon dioxide emissions. The greenhouse gas emissions is equivalent to 5.5 kg carbon dioxide for each kilogram of methyl methacrylate. So in any measures, you can say that this process, it's impossible to be industrial. But what we can say is, don't try this at home. For production of methacrylic acid, we have integrated bio and chemical catalysis. And in this process, we started with growing first gluconobacter oxidant cells, acetic acid bacteria. And then we use it as a whole cell biocatalyst for conversion of 2-methyl-1,3-butanediol, which is an industrial byproduct, to 3-hydroxy-2-methyl-propionic acid. And this process is mediated by 2 enzymes, alcohol dehydrogenase and aldehyde dehydrogenase. Later on, the final compound, we do a dehydration using titanium dioxide at 210 degrees centigrade, and we can end up in methacrylic acid. All the reaction is done in water, so you have highly pure product. So this is the biological step, and the second step is the chemical step. And we can see here the conversion of 7.5 milligram per milliliter of substrate using 2.6 milligram of gluconobacter oxidant cells. We can achieve 100% conversion almost in 24 hours. At 5 gram substrate gram per liter, you can convert in around 10 hours. The catalytic dehydration step, we found that the optimum condition is around 210 degrees centigrade, and we can achieve high conversion and quite high yield. And we can see here the starting material, that's the 2-methyl-1,3-butanediol. And during the conversion, we can find this aldehyde is also accumulated here to a small concentration and the formation of the product. And then in the third, this is the final product, 3-hydroxy-2-methyl propionic acid. And after catalytic dehydration, we can end up in methacrylic acid. So it's quite a promising process. And now I end up with the concluding remarks. First, choice of the product is very essential and strategic for development of biorefineries. Starting cheap, ending cheap. So you have to properly choose the starting materials before you start the reaction. High cell density processes such as fermentation or biocatalysis, it can yield high volumetric productivity and titers, and I think this will have, the future is for high cell density fermentations and biotransformation. In situ product removal and complexation, this seems to be crucial and essential if you are handling toxic or inhibitory products. Wholesale biocatalysis, it's very advantageous if you have a number of enzymes even, is a promising technique, and especially if you have a multi-step enzymatic reaction and if you have a cofactor recycling, so you don't need to use any nitrogen source. And finally, we should make use of the accumulated knowledge in chemical industry and use it for supporting the industrial biotechnology. And we show here one of examples using integrated refineries. Future perspective for propionic acid, we think that a combination of different techniques, we have been focusing on fermentation, but if we can combine it with in situ recovery, we can reach the industrial requirement. Also, the downstream processing is quite problematic, and it needs a lot of work to get it quite cheap. For 3-hydroxypropionaldehyde, we need to do further optimization of the in situ complexation and removal using bisulfite. We sought to connect several columns so we can keep the process for a longer time. And also 3-hydroxypropionic acid, we need to work more for enhancing the productivity and concentration. And also we have some plans for metabolic engineering to get only 3-hydroxypropionic acid as a final product, complete conversion of glycerol to 3-hydroxypropionic acid. Regarding methacrylic acid, we can take the industrial process for acetic acid production as a model, and we can try to reach the same level of this process. And finally, I would like to acknowledge, thank Vinova for financing the project, and my supervisor, Professor Rajni, and Associate Professor Sang Hyun, and Dr. Per Person for their supervision, and also the co-workers, Roya and Teresa, and members of the Department of Biotechnology for making the nice environment, and also the project members from different companies. It's a lot of names that I have to say, but from Vinova and from Perstor, Liquidy and Bonashi, Danisco, Metabolic Energy, Cambrex and GE Healthcare, and finally from London University. So I would like to thank everyone, and a small tip here that sometimes you need to look at life from a different perspective, and you can make this perspective as a biorefinery perspective. Thank you.
That's okay.
Okay, thank you very much.